Hello friends, welcome back to our YouTube channel Nesting Padasali. So in this video, I'll be discussing the answers for GATE Biotechnology 2022 Life Sciences. That also in this video, I'll be discussing the answers for Biochemistry. If you had selected Biochemistry as one of your section, please clarify your answers with mine. Okay, so that we can cross check any answer so that once the final answer key is released, we can help the student who are willing to challenge the question. So the first question, which of the following compound is not a translational inhibitor? So these inhibitor, they inhibit the process of translation, which is other name called as protein synthesis. So first, chloramphenicol is a correct answer. Yes, chloramphenicol is a widely used protein synthesis inhibitor. Next is cyclohexamide. Yes, the cyclohexamide is used as a prokaryotic translation inhibitor, and this puromycin. So this puromycin is nothing but is the amino nucleoside antibiotic, which means they will involved in causing premature chain termination during the process of translation. Okay, so this puromycin, all the components are correct, whereas this rifamycin is the odd one out. So this is a MSQ type of question, and the correct options are chloramphenicol, cyclohexamide, and puromycin. Whereas this rifamycin is not a translation inhibitor because it is a type of transcription inhibitor. So they will inhibit the bacterial DNA-dependent RNA polymerase. So they don't involve in inhibiting the process of translation. So the correct option are chloramphenicol, cyclohexamide, and puromycin. Next question, which of the following statement about IgA antibody is correct? Okay, so this is again MSQ type of question. So it is secreted in colostrum of milk. Yes, it is a correct option, and it is transported across a cell by transcytosis. Transcytosis is nothing but it's a type of transcellular movement, friends. So it is allow the component inside the cell to move from one location to another location by forming vesicle. Okay, so this option 2 is also correct, and its secretion is facilitated by poly-Ig receptor. So this poly-Ig receptor is nothing but it's a polymeric immunoglobulin receptor, and it is involved. This receptor is helpful in the process of transcytosis. Okay, so it is only helpful for transcytosis process of IgA antibody and IgM antibody. So this option is also correct. It is primarily existed as dimer syndrome. Yes, this IgA is the only antibody which is acting as a dimer. So all the 4 options are correct. Which of the following carbohydrate have or has a beta-1,4 glycosidic linkage? So it is a MSQ type of question. If you are given a correct option, then 2 mark will be given, and if you given a wrong option, then no mark will be detected because they won't reduce the mark because it is there is no negative marking for MSQ type of question. And the correct option are your cellulose, chitin, and lactose, because maltose is again form a alpha-1,4 glycosidic linkage, whereas the cellulose is forming a beta-1,4 glycosidic linkage, whereas this chitin will form a beta-1,4 glycosidic linkage, and this lactose will form a beta-1,4 glycosidic linkage between your galactose and glucose. Next thing, which of the following enzyme is located outer mitochondrial membrane? Okay, I think many of the students are getting confused. So except this monoamide oxidase, all the enzymes like your citrate synthetase, fumarase, and succinate dehydrogenase all are involved in the process of respiratory chain or electron transport chain. So that circumstances, all these compounds, all these enzymes are present in outer mitochondrial membrane. Whereas this monoamide oxidase is nothing but is involved in oxidizing monoamide, that is neurotransmitter, and they are present in submitochondrial membrane, that is they are generally present in inner mitochondrial membrane. And you can refer the paper published on 1967 under JCB. Okay, though the author name is Erwin, you can refer that paper for your further references. So the correct options are citrate synthetase, fumarase, and succinate dehydrogenase. Next option is that is again an easy question. Dye was allowed to undergo migration chromatographic paper. So here they are speaking about paper chromatography, and the dye and the solvent front are migrated as 5 cm and 3 cm respectively. And they are asking you to find what is the retention factor. So the retention factor is nothing but is defined as a ratio of distance moved by the solute. So in this circumstances, they are speaking about distance moved by the dye, which is 5 cm, and the distance moved by the solvent. Okay, so if you had made them opposite, means then the entire thing will be wrong. So the RF value is given by 5, which is the distance moved by dye, divided by distance moved by the solvent, that is 30. So the option correct answer is 0.25. It is a numerical answering type. Next question, pKa value of carboxylic acid and amino group of amino acid with a non-ionizing chain are given. What is the isoelectric point? So the formula to find out isoelectric point is Pi is equal to pKa 1 plus pKa 2 divided by 2. So here pKa1, that is carboxylic acid, is given as 2.17, and pKa2, that is pK of amino group of amino acid, is 9.13 divided by 2. So the correct option of that is isoelectric point of this particular amino acid is 5.65. They didn't mention any amino acid name. Which of the immune cell listed below are a granular? So you should differentiate. Leukocytes are classified into two types. One is granular, which contains whose cytoplasm consists of granules, and another one is a granular. A granular leukocytes is nothing but they lack granules in cytoplasm. So there are two types of leukocytes that are classified as A granular. One is your lymphocyte and another one is a monocyte. Okay, so lymphocyte means your monocyte, and lymphocyte is nothing but T cells and your B lymphocyte. T lymphocytes and B lymphocytes. So correct options are RNS. Whereas this eosinophil, mast cell, basophil, neutrophil are belong to granular type of nucleocyte. Okay, they consist of granular, so that's why they are easy to differentiate by addition of eosin dye. Next thing, number of ATP molecule required for complete acylation, only one molecule of carbon dioxide. They are asking what is the molecule of ATP that are generated by assimilation of one molecule of carbon dioxide in Calvin cycle. So the question is a direct question, and there are 3 ATP molecules that are required to completely assimilate only one molecule of carbon dioxide in Calvin cycle. Next thing is a problematic type of question. The absorbance of 5 into 10 to the power of 4 molar solution of tyrosine at 280 nanometer wavelength is 0.75. The path length of cuvette is 1 centimeter. The molar absorption coefficient is what? So this is a formula. A is equal to epsilon Cl. A is absorbent, epsilon is a molar absorption coefficient, and C is a molar concentration, and L is a path length. So first you need to apply all the value. Absorbance is 0.75 and concentration is 5 into 10 to the power 4, and your length is 1 centimeter. So after applying, if you rearrange, the molar absorption coefficient is 1500. Okay, so you need to round up. Next question, which of the following statement about DNA polymerase 3 of E. coli is not correct? This is the easiest question, friends. So DNA polymerase 3 is a holoenzyme that is involved in prokaryotic DNA replication, and this particular enzyme was first identified by Korenberg. Okay, so first thing is that DNA polymerase catalyze Nick transition. No, DNA polymerase of course, but DNA polymerase 3 is not involved. DNA polymerase 1 is involved in catalyzing the Nick translation. Nick translation is used in the process of labeling of DNA. So option A is wrong. Next is absent is lethal to E. coli. Yes, because this DNA 3 polymerase 3 is involved in replication of E. coli. If the DNA polymerase 3 enzyme is not present, then ultimately replication in E. coli will not be happen, and the E. coli will not be multiplied. So it will lead to lethal cases in E. coli. Next, this DNA polymerase 3 is involved in synthesizing a complement DNA strand using a SS template. Yes, they will use a single strand template to synthesize a complementary strand, and this DNA polymerase 3 is also involved in processing 3' to 5' hexonuclease activity, which is nothing but proofreading activity. Okay. So, which of the following, next question is, which of the following statement about endogenous synthesis of insulin is correct? Okay, so this is a MCQ type of question. So, insulin is synthesized as pre-proinsulin. Yes, correct option. And once this pre-proinsulin synthesized, it will be converted into proinsulin. Yes, it is also a correct option. Next thing, single site cleavage of proinsulin eliminates C chain. No insulin, if you see the structure, there is no C chain and all. That is only A and B chain. So, option number R alone is wrong. Whereas if you see option number yes, mature insulin consists of disulfide linkage between A chain and B chain. So PQS. Okay, so these three options are only correct. Which of the following enzyme converts testosterone to estrogen? So I don't know whether this question is a MCQ type of question or MSQ type of question, but the major enzyme involved in converting your testosterone into estradiol is your aromatase enzyme. So this aromatase enzyme play a vital role in converting your testosterone into estrogen. Next question, purification of 6-histidine tagged protein using nickel. NTA is nothing but nitroacetic acid. So this is a chelating compound. This acid is used to chelate the nickel ion. So example of your affinity chromatography. Yes, this nickel NTA is used as an affinity chromatographic matrix for purifying the recombinant protein that are carrying your 6-histidine tag. Okay. And next thing is a problem type of question. Your 40 time dilute sample of single stranded RNA gave A260 value of 0.01. So they are diluting the sample to 40 times. The concentration of single stranded RNA before diluting of microgram per ml is. So this thing I think you should mark up every time when you go for examination. Okay, if you are going to write a question paper for CSAR, then you should know this particular formula. So better take a note in your rough notebook, or you can take a picture. So for 1 centimeter path length, the optical density at 260 nanometer equal to following solution. So here they are speaking about a single strand RNA. So here only RNA is there. So for single stranded RNA, that is 40 microgram per ml. So you need to simply multiply 40 into the value 0.01. Again they are diluting the sample to 40 times, and once again you need to multiply the value to 40. This 40 is nothing but 40 microgram per ml of RNA is present, and this 0.01 is a value given over here, and this 40 signifies 40 times they are diluting, and the answer is 16 microgram per ml. And next video I will be discussing the answers for zoology. Thank you friends. Thanks for watching.